Hello, everybody. It's my pleasure to introduce for today's talk my colleague from the University of Ljubljana, Boštjan Gabrovšek. Boštjan is partly employed by the university, by the Faculty of Education, and as an assistant professor, and partly as a researcher at a research facility in the south of Slovenia in Novo Mesto, which is not so far because Slovenia is really very small. So Boštjan is going to talk to us today about topological analysis of knotted proteins. So please, Boštjan.
Okay, thank you very much. Also, I would like to thank the organizers for inviting me to give this talk. Okay, so the plan for today's talk is to talk a little about different aspects of analyzing knotted proteins by kind of their topological structure and geometric shape. So the plan is, first I will talk just briefly to define what are knots, what is protein folding, what are knotted proteins. So we have different kind of different ideas what a knot can be inside a protein, and I will try to give a few examples. And then the remaining of the talk will be about how to detect knotted structures inside proteins. So first will be purely mathematical invariants. They have a couple of downsides in this kind of real world scenario. Then I will talk about a bit of our project. I'm using persistent homology, how to analyze certain types of knotted proteins. I'm not sure if I will have time to talk about the last part about using machine learning to detect knots, but we'll see how much time I have. Okay, so to start, just briefly to give the topological kind of basics is a mathematical knot is just an embedding of a circle into the three-dimensional Euclidean space, or sometimes we want to embed it in the three-sphere. The definitions are very equivalent. So usually we talk about something to be knotted if it is at least a trefoil. So there exists a whole table of knots. So we have the unknot, so a circle that's not knotted, and then we have different types of knot. And usually we donate a knot by two numbers. So the number of minimal crossings it has in all possible projections or in all possible configurations. So there's always a minimal number of crossings in the projection. And then we have the index. So for example, the trefoil knot has 3 crossings and it's the only one of its kind. And then, for example, with 5 crossings, we have 2 candidates. So we have 5-1 and 5-2. And then we will also talk a bit about links. So links is basically just if you have several circles embedded in R3. So we have the unlink, the Hopf link, the Salomon's knot, probably link, Whitehead link, and so on. So knots appear in nature in various contexts. So for example, we can find them in nature, industry, fluid dynamics is also interesting. Of course, sailing, DNA, chemistry, molecular knots. These molecular knots is an interesting project that I think it was done some years ago by a Slovenian team where they constructed the knot table up to some amount of crossings, and they constructed the smallest knot, artificially created knots. I think there were only some nanometers big. So they used some special techniques and they got all knots up to, I don't know, I think 9 or something crossings. Of course, we have in robotics, sometimes configuration spaces appear as knots. You have, of course, knots in arts, electromagnetism, and today we'll mostly talk about knots in proteins. Just a very brief history. So the whole idea of something being knotted and studied systematically began with Tite in 1876. He constructed a knot table up to 7 crossings. And he gave, these are more, not really knots, they're kind of the pre-attempt of a knot theory, let's say, where he classified just purely projections of knots. So let's say just combinatorial diagrams. And then over the years, people were constructing knot tables up to many, many crossings. So it started with 10 crossings. with many errors, and then people made these knot tables bigger and bigger and bigger and containing less and less errors. So, for example, today we mostly use this Rolfsen knot table, but it contains one error, but we have identified it. So he thought he created a knot table up to 10 crossings. It contains, if I'm not mistaken, a few thousand knots. I'm not completely sure, but there was one error which was then solved by Perco, so it's known as the Perco pair. And now we have knot table up to 19 crossings, and it contains 350 million knots. So now I think, I'm not sure if anybody is continuing this work. I think people, it's also this knot table it's very large. It's a few terabytes to download. So I'm not sure if anybody's working on increasing this, because I'm not sure how much sense it makes anymore. But anyways, it's a possibility. So this is the existing knot table. So we have a knot with 3 crossings, the figure 8 knot with 4 crossings, and so on. And we know that the number of knots given a certain number of crossings, it grows exponentially. So knot tables are exponentially coming bigger with the number of crossings. And now I will talk a bit about proteins, and then I will join the 2 subjects. So proteins are large molecular chains, also called biopolymers, composed of dozens, hundreds, and even thousands of amino acids. So you have kind of these 4 structures. So the primary structure is just a sequence, purely linear sequence of amino acids. And we know that this naturally forms into a kind of 3D structure. And proteins, it's basically they're the building blocks of life. So we find them, so we have so their function is, let's say, structural support, collagen, keratin, enzymic activity, transport, storage, signaling, defense, movement, regulation. So it supports all kind of building blocks of life. And the important fact basically here is that its native state, so its 3D structure, is determined purely on the amino acid sequence. So given just a linear sequence, we should be able to uniquely construct the 3D structure. Of course, there are a handful of examples where also you have, for example, there are proteins that change their shape doing some function, or they can be misfolded, for example, for some in some types of diseases, but these are more exceptions than a rule. And then we have the folding process. So proteins fold from a linear chain, so they are generated as a linear chain, and then they go into a stable 3D structure. And of course, we call this transition from the unfolded state to the native 3D structure. It has to go through this energy landscape. And of course, it wants to minimize its energy to come to the native state. And we know that it has to very often, of course, some of them have a direct local minima, so it's a very fast folding, but often they have to go through this energetic barrier. So they have to go into so for example, especially for knotted proteins, they usually have to go through this energetic barrier. So they have a minimal energy, they have to increase the energy to go into some topological configuration, and then they can go to even further minimal energy. And of course, misfolding and aggregation can occur, and the proteins get stuck into a non-native state, leading into dysfunction and diseases. So, for example, Alzheimer's disease, Parkinson's, cystic fibrosis, prion disease are all diseases that are caused by protein misfolding for one reason or another. Let's go now to knots in protein. So it was for many years believed that nature could not tie a protein because of these kinetic challenges, especially in these high activation barriers it has to pass through when going through the energy landscape. So there would be a high activation barrier of such complex structure, and it would not kind of pass through the evolutionary filter. And then Mansfeld in 1994 suggested that proteins perhaps can be knotted, and he found a candidate of a protein which he suspected that that would contain the trefoil knot, so this kind of the most trivial, so the least complicated knot that's not the unknot. And back then, people didn't really believe him. They thought it was ridiculous. So here are some quotes that he received after making it public that he is trying to find a knotted protein. And 3 months later, after that announcement, Mislow confirmed the existence of the 3-1 knot exactly on the candidate he suggested. And then a few years later, people started to find more and more complicated knots inside proteins. So not just the trefoil, but also the figure-8 knot, the 5-1 knot, and so on. And in 2000, Taylor found that also knots can be deeply knotted, which was also kind of a new revolution because all the previous knots were very shallow. By shallow, we mean that if you just cut out a few amino acids from one end, it becomes unknotted. So for example, this is a trefoil. If you would cut out just a few amino acids, it would not become a trefoil anymore. And these knots that were found by Mislow and others, they were all very, very shallow. So just a few amino acids, I think, would make it an unknot. And he found a deep knot. And by today, how we define a deep knot is that if we cut 30% of amino acids from one end and 30% of amino acids from the other end, that it's still a knot. So that's today's standard definition of what a deep knot is, which means that it's contained in the middle 40%. So we call this part that actually forms a knot core. So it means that the knot core is inside the 40% middle part. And by a recent study, we now know that almost 80% of all knotted proteins are indeed deeply knotted. So that was done by a systematic study of AlphaFold, which I will talk, I think, on the next slide or the one after. So it's quite interesting that we have mostly deep knots. And so one might ask if these knots are just random or they have some function or some other property. And it's mostly believed that the knots are there for a reason. And one very, very interesting case is the case of the human ubiquitin hydrolase. It's a protein that has a relatively complex knot, 5-1. So it has 5 crossings. It looks like this. And its function is that it rescues proteins from degradation. So basically, the proteasome, it's a mechanism inside the body that degrades protein. So if a protein is, let's say, corrupted or it's not longer needed, then it gets these ubiquitin markers on it. And these markers then are attracted through this proteasome, and it's basically pulled inside, and this guy then degrades it into smaller pieces, which are then reused. And often it happens that some proteins are marked for degradation, although they shouldn't be. And this ubiquitin hydrolase, it rescues such proteins from degradation, and which means that it's often found this protein with this ubiquitin hydrolase, it's often found in the proximity of the proteasome. And the 5-1 knot, it suggested that it prevents the knot from being pulled inside because it's quite a compact, deeply knotted, it's quite a compact knot, and yeah, like I said, the knot gets, it prevents the protein from being pulled. So it has kind of this structural, so the knot itself has a structural advantage. And actually there was a Nobel Prize for the discovery of this ubiquitin mediated degradation, so how this system works of protein degradation. And just a fun fact, our brain contains 2% by mass of ubiquitin hydrolase, which means that 2% of our brain contains knots. Okay, so the next step is how to go back a bit to mathematics. So how do we actually classify what a knotted protein is? Because we know that in mathematics, a knot is a closed circle embedded in 3 space, and proteins have open ends, the vast majority of them. And we use basically in general, it's still an open question if how to kind of uniquely close a protein, but 2 methods were used in the past. So kind of the first methods that were kind of introduced were some sort of direct closure. So either you can just have the 2 ends, so you have an amino end and an acid end. You can close them by a direct segment. Of course, this isn't a stable operation because you can just permute one end a bit and you can get a different knot. The same thing happens with closing it by the mass center. So take the mass center and close it. It's also not a stable type of closure. And then you can just, for example, you can project from the mass center you can put the protein inside a ball, project the center. So take the line from the center to the one end, get the intersection, and to the other end, take the minimal curve, and you can get another type of closure. So there's many ways of a direct closure. But nowadays we usually use these stochastic closures, so probabilistic closures, which basically you take a protein inside a big 3-ball and then take many points. So usually we take around 1,000 or 2,000 points on the surface randomly, and just either you have a closure with 1 point, so you connect the endpoints to this 1 point, or connect them take 2 random points and connect them by taking this segment, and the geodetic. And at the end, you can get different types of knots, of course, depending on how you chose to do points, and then we form these atlases or maps of knots. So for each, so this I think it's a 1-point closure. So for each point on the sphere, you can then compute what type of knot you get, and if there is one type of knot that kind of is in the majority, you can say that it's with a high probability knotted at 6-1. But actually it's not usually this complicated, because the endpoints of the amino acids are often very close to the convex hull of the protein. So they're usually not somewhere in the center of the protein, but they often are you often have something very complex, and then you have 2 tails that stick out. So in the majority of cases, this closure is not it's not a big problem to determine. It's not a type. And then, of course, if you do some new methods of detecting proteins and so on, so for example, one that we will present today using persistent homology, you usually go to some big data approach. So you have to have many examples, see how well your method does, how fast it is. So, and there's basically two databases where you can take 3D structures of proteins. So one is the PDB, so Protein Data Bank, which are experimental determined structures. So either today, more or less, they use X-ray crystallography, and it contains approximately 200,000 structures. But a couple of years ago, there was a big revolution, because now Google, so DeepMind, so it's a company, I think a daughter company of Google, created AlphaFold, which is an AI-based prediction, and basically it takes as an input the sequence of amino acids, and it can, with a high probability, predict what the 3D structure is. And here we have 1,000 times more structures. These numbers are a couple of years old, so it might be even more now. And which means that just in a few years, we had 1,000 more examples to study if you want to study, let's say, finding knots and so on inside proteins. So there's a lot of work going on now in this direction by taking the data from AlphaFold. So the AI predicted 3D structure, because in general, it's a very difficult thing. So we know we know what the amino acid sequence is from proteins, but to determine the 3D structure, it's quite a complicated process. So either you do it experimentally or with AI. And when AlphaFold developed its AI engine, it was a very big revolution. They called it the solution to the 50-year-old research problem, because experimental determining the 3D structure, it's a very, very expensive project. So for one protein, it can be tens of thousands of euros to determine its 3D structure, and we know that we have 200,000 structures determined. And also, there were problems, of course, in this AI, because a typical protein has 10 to the power of 300 different configuration, which is a lot. And of course, this AI made quite a revolution to pinpoint the one single configuration. So these are just results historically. So for years and years, people try to develop some sort of AI or some other software to determine the 3D structure of a protein given its sequence of amino acids. And the success rate, so this is the quality. It's not exactly a percentage, but it's some index, which is, you can think of it as a percentage how good the prediction is. And for every 2 years, there were these CASP competitions when people try to improve their software to predict the 3D structure. And we can see that over the years, not much progress was made, but then AlphaFold kind of made a revolution. So in 2020, there was AlphaFold 2. And this 90% barrier is a barrier that is considered roughly the equivalent of experimentally determining the 3D structure. So AlphaFold 2 become almost as good as doing experimentally determining the 3D structure, so using X-ray crystallography. And now we have AlphaFold 3, but I couldn't find what the results are, because they are kind of keeping things closed source. And those CAS competitions are no longer active as far as I know. So I think it's this AlphaFold 3 is now, it's relatively new. So we still don't know if they reach the barrier of experimentally determined structure or not. So the release AlphaFold, the code is released for old versions, but the new versions, they kind of keep closed source. So maybe the barrier is reached now, but I am not sure. Okay, so we were talking about knots in proteins. So this is kind of the standard way of saying a knot is something is knotted or not. So we can think of a protein, if it contains, let's say, a trefoil or a figure-8 knot, you can call it a knotted protein. But we can also have different classifications or meaning what something knotted is. So for example, we have a slip knot, which is in a way also knotted. If you would cut, oh, this strand should go over, the red one should go over the blue one. There's a bit of a mistake in the picture. So for example, if you can imagine pulling the blue part and the red part, this would trivialize, taking into consideration that this red part goes over. But if you would cut the protein here, it would be knotted. So something like this that is topologically trivial if you would pull out the ends, but it's still knotted. If you cut the protein, we consider a slip knot. Then we have a lasso, which means that, as we'll see a bit later, proteins can also have these intramolecular chains, which we will call bonds. And if we have a protein chain and we have this bond, it forms a disk. And if some other tail of the protein pierces this disk, we call it a lasso. And this is also topologically not trivial in a way, and it also has to go through this high activation barrier, because it's very difficult if you have let's say a limited area piercing through with another strand. You have to reach some activation barrier, so they're also very interesting to study. And then we can study also theta curves, so we have knots or even unknots with added bonds. And we can also study links. So deterministic links are links where you also have these bonds, and they form, for example, this forms the Hoff link if you ignore the tails. I'm not sure what this 8% means. I think that this 8% is referring to lasso proteins. I think there's a bit of a mistake, but we know that from AlphaFold now, so the big database with 200 million structures, there's somewhat less than 1% of proteins are knotted, which still is a big number considering how many proteins there exist. So I have this graph, it's from a paper of Joanna Sulkowska, where they studied how many knotted proteins do organisms have containing on their family, so bacteria, fungi, plant, animal. We see that fungi have a bit more knots in general, but also animal are quite knotted, so yeah. And we know for a fact that every living organism contains proteins because you have some proteins that are shared among living organisms, and we know that for big families that all families contain certain proteins, and we know that some of those proteins are knotted, so it's been shown that all living beings have knots. But now if we consider that also we take into account that something is knotted if it doesn't have a trivial structure, including bonds, we have a much richer kind of this notion of what something knotted is. It means that it's not just a simple theta curve, so just a circle with a bond, but if we take into consideration that we also have these intermolecular bonds, we can approximately say that 20% of proteins are entangled. Although the backbone, as in this case, is a trivial knot, you cannot trivialize it if you have some bonds inside. And among proteins that have bonds, they have in average 2.6 bonds. So proteins have usually around 2 or 3 bonds, and those structures are usually not topologically trivial. Just to see how many proteins have been found so far. So this is a very ongoing process. So I think that every, let's say, half a year, somebody finds a new knotted protein. So I'm not. This is probably not the full table, but for a while, we have found the 3-4 knot, the 4-1 knot, the 5-2 knot, the 6-1 knot. And then lately, with the development of AlphaFold, more complicated knots were found. So these first 4 were found just using the standard protein data bank, using this experimentally determined structure. But with AlphaFold, people found more and more complex knots. So very lately, I think this 8-3 was found. Then we have 6-3. Then we even have a connected sum, which means that we have 2 trefoils inside one protein. And then we have this nice 5-1 and 7-1 knots. And these are not just AI predicted, but the AI helped to predict it. But of course, they have to crystallize it and do the classical experimental determination that there really is a knot. So these have been confirmed even experimentally and with AI. So first with AI, then they synthesize the knot and confirm it again. And probably there are a few more candidates these days. I'm pretty sure there are. Okay, so how to classify such structures? So we have different types of what something knotted might mean. And of course, we are mathematically interesting how to if you have a 3D structure, so just a sequence of atoms in 3D space, how to determine if the thing contains a knot or not. So one way is just use mathematical invariants. Then we have this nice experiment with using persistent homology, which I will talk, and also you can use ML. Which I don't think I will have time to speak about. Okay, so the classical way, kind of the most natural way how to detect if a knot is if a certain curve in space is knotted, you can project it, obtain a knot diagram, so just a projection, and you can use some mathematical invariants, so invariants from knot theory to compute some value, which is of course invariant under ambient isotopy. So if you move it around, create new crossings, and it's topologically the same, it returns the same invariant. And probably one of the most famous polynomials is the Jones polynomial, or it can be just by some substitution converted to the bracket polynomial. So you can create, basically you give a knot diagram and you have these three rules that give you some polynomial value. So just the value on the unknot is 1. If you have a crossing, you can smoothen it on 2 ways. So you can put, so if you take the over strand and you rotate it in a positive direction, it uniquely cuts this into 2 strands. So you can remove a crossing by having one smoothening. The other smoothening, you take a times one smoothening plus b times a to the power of minus 1 times the other smoothening. And then you can just take a crossing, you smoothen it, you get expressions, and you can recursively do this until you end up just with a bunch of unknots. And we have this last relation which tells you if you have an unknot and a disjoint union with something else, you can multiply it by this expression. And we know for a fact that this is an invariant. So there is a purely mathematical way how to compute if something is knotted. So if the bracket polynomial will be 1, we know it's very likely unknotted. This is also an open question in mathematics. If the Jones polynomial can detect the unknot, and we don't know. So there people in general, I think, believe that it can detect. So only the unknot has value 1, but it's not proven. But since we have tested all knots up to 19 crossings, none of them have value 1 if they are knotted. So there's a almost certain you can get the knot type from this polynomial value. Then the second approach is to just use systematically Reidemeister moves. So we know that it's a theorem from Reidemeister that if you have a knot diagram and if you can only use these 3 rules to simplify every knot if a simplification is possible. So the theorem says that 2 knot diagrams are equivalent, so ambient isotopic if and only if they can be so one can be converted to another using these 3 Reidemeister moves. So we have this Reidemeister move which either creates a loop or removes a loop. We can pass one strand over the other, or we can pass a strand through a crossing. So for example, this is a sequence. So for example, this one, we don't immediately see that it's topologically trivial, but we are able to do a sequence of Reidemeister moves to create the unknot. Okay. And of course, this since these invariants are quite difficult to compute, for example, if you have, because let's say you have a protein, proteins are in general very geometrically complex structures. So usually you do not want to compute the, let's say, Jones polynomial or the bracket polynomial immediately, because for each crossing you get 2 other candidates. So it's MP hard, so it's 2 to the power of number 2. So the complexity is 2 to the power of number of crossings. So first you want to apply on some smart way a sequence of Reidemeister moves to reduce it, and then you can compute the polynomial. So that's kind of the usual way. And we are also doing a library that does that, also not only for knots, but also for all kind of other structures. So example, planar graphs and so on. Now I want to talk about also lassos, because that's the part we are working in the persistent homology. So lassos, like we said before, are knots that also have these intramolecular bonds. So first, let us see what types of bonds we have. So protein bonds. So peptide bonds are covalent bonds that link amino acids together to form a protein's backbone. So this kind of backbone of the protein is formed by peptide bonds, and between them you can have non-covalent bonds. So these are hydrogen bonds, disulfide bridges, ionic bonds, van der Waals forces, hydrophobic interactions, and so on. And they are used to stabilize the protein. Of course, they're not so strong as the backbone, but they're still significant. They are still very important in the protein 3D structure. So we will study from now on proteins with bonds. So there has been a relatively recent analysis of just theta curves inside proteins. So this was a paper done by Joanna Solkowska and her students. So, and they identified 7 different types of theta curves inside the protein data bank. So that is not considering the AlphaFold. And I'm sure that considering the AlphaFold, we would have many, many more candidates because we have 1,000 times more structures. So we have non-trivial theta curves inside the protein. And a theta curve is, I probably haven't defined it, so you can define it just as a 3-valent graph embedded in 3 space. So you take a graph with 2 vertices, 3 parallel edges, and you embed it in 3 space. So it looks like the letter theta. So this was a nice paper quite recently published. Now the problem we are really facing is that if you try to generalize these, let's say the bracket polynomial to something, it can be generalized to something that that is able to detect also different structures like bonded knots or theta curves. Most invariants pretty much fail. I'm not sure about the exact reason, but if you take, for example, if you compute the topological version, so if you generalize this bracket polynomial to a bracket polynomial of a bonded knot, and you take the topological version, which means that you can also freely kind of rotate stuff around the vertex. So the vertex is not fixed, but you can move around also the endpoint, the kind of edges emanating from the point. You get a topological version of a bonded knot. And if you compute the Kaufman bracket of this topological version, or even the Humphrey bracket, the Humphrey polynomial of this, it is trivial. It does not tell you anything about the configuration of the bonds. And I think the underlying reason is that most topological invariants of knots depend on the complement of the knot. So really you can think about it as a thickened knot. So if you take the complement of the knot, usually these invariants are constructed in theory of the complement, which means that we study kind of a thickened version of the knot. And we know that topologically something, if you thicken this handcuff, for example, you can unlink it. So really you lose, by thickening a knot, you lose a lot of topological or combinatorial information about the knot. So here, I think this is a video which shows that it's this old handcuff trick that topologically you can always unlink a handcuff. But in knot theory, since we are dealing with one-dimensional graphs, these should not be the same. So we want to have an invariant that would detect this. And these old invariants based on classical knot theory usually fail in the topological case. There is one exception, and that's the Yamada polynomial. This is a polynomial that it was constructed by Yamada, and it can successfully detect, just looking how much time I have, it can successfully detect also these bonded knots, let's say. And it is kind of a combination of invariants from topology and invariants from graph theory. So it combines a bit of topology and a bit of combinatorial data. So it's defined recursively similar to the Kaufman bracket, but we see that some part of the definition corresponds to the Kaufman bracket, and then you also have certain recursive relations that are connected to graph theory. So for example, the chromatic polynomial, the Tute polynomial have this relation. If you have an edge, you can contract it and you can delete it and you get 2 summons and so on. So it's a combination of invariants, and it's quite successfully can detect type. It can detect the ambient isotopy of bonded of embedded graphs. But the problem is that its time complexity is very inconvenient, because it has O to the 3 to the power m, because for each crossing you get 3 summons, and it's very impractical to use. So for example, if you had a protein with 100 crossings, it would be 3 to the power of 100, which is almost impossible, which is impractical, impossible to compute. So as soon as n is, let's say, greater than 20 or something like that, you have a, it's almost impossible to compute this Yamada. So there's still here an area where we have millions and millions of proteins, their 3D structure, but we are unable to really compute their, let's say, bonded knot type, or if you look at them as graphs. So that's still an open question how to do this successfully. Which kind of, we are headed into our next step. So I will now, in the remaining of my time, talk a bit about an experiment we did with Giga Wirk, Joanna Sulkowska, Paula Kovicchioli, also Bartosz Gren. We try to use persistent homology to detect some type of knotted structure. So in particular, we were interested in lasso proteins. So lasso proteins are a class of proteins which part of the polypeptide chain threads through a covalently closed loop. So here you have the image. So we have this, I can find my mouse cursor. Okay, on the left, we can see the protein, the backbone, and we see that it forms a closed loop by this disulfide bridge. And part of the tail then pierces this closed disc. And there are many ways of how this can occur. So the simplest piercings, also the simplest lasso protein that exists is L1, which just means that the tail pierces this disc once, it can pierce it twice. So we have L2, L3, and so on. And if it pierces in a way that it also rotates around the backbone, we have so-called supercoiling. So at the bottom you have, at the bottom you have a lasso LS2, which means a supercoiled lasso with two piercings through the minimal surface. I mean, this surface bounded by the protein. Then we have a double coiled, then we have a supercoiling of degree 3, and so on. And with AlphaFold, they found much more complicated lassos. So I think it goes up to, I'm not sure, 20 or 30 or something like that, piercing through the minimal surface. So there's really a big database of these lasso proteins. So why does it matter to study lasso proteins? So we know that they are thermal and kinetically stable due to their kind of locked topology. Many have antimicrobial, antiviral, or anti-cancer activity, and they can serve as a scaffold also for drug design, since the threaded topology is rigid and chemically tunable. And we know that from LassoProt, which is basically a database on the website developed by if I'm not mistaken, Joanna Sulkowska and her team, we know that 50% of lassos are non-trivially looped. So if you take proteins that have these bonds and they form a disk, 50% of them are non-trivially looped, which means that they kind of pierce, so they have this supercoiling and so on. Now, the traditional approach involves computing the minimal surface. So how to compute if a given protein is a lasso protein? So basically, you have to compute the minimal surface, and then just look if the tail pierces it. So it's quite a computationally heavy thing to do. So computing, of course, the minimal surface is not trivial. And then also you get, so some proteins are kind of very direct. You can see that they are lasso. Then you have medium, and then a lot of them are just very complicated. So this method can be slow. So that's why we wanted to turn to persistent homology to have an alternative method. And in theory, in some cases, also this surface is not well defined. At least it's not a stable operation, because we know that certain curves in space have two different minimal surfaces. Although I don't completely understand the way they computed the surfaces, but as far as I talked to the team, they took this stability somehow into account. So it's not completely unstable, their methods neither. So it's quite a very, very nice database, and a lot of work went through into computing all lassos with this high number, with this very geometrically complex structure. So it's a very nice, it's also a very interesting website to look about, like how complicated structures are, and it's a really amazing webpage. Anyway, so to continue, so how to kind of compute this? So we would like to have a method not using minimal surface to compute if a protein has a lasso structure. And the persistent homology is kind of very, very nice to do. So just to give just a very brief introduction to persistent homology. So basically, I'm not a very big expert on persistent homology, but still, the idea is to just start with a set of points, which in our cases will be just atoms, positions of atoms. And then you choose a scale parameter, so a radius r, and then you can you make r bigger and bigger, which kind of goes into this you say that you choose bigger and bigger scales, and if then and then you create based on some radius, you can create a simplicial complex by just connecting 2 points if their distance is less than r. So if their let's say ball around them r is connected, and of course you have Vietoris strips complexes and chess complexes. So Vietoris, the one we chose was Vietoris strips because it's easier to compute, which basically means if you have 3 points connected, so it's kind of a clique complex. So if you have, let's say, 3 points connected, then you put in a triangle. If you have 4 points connected, you automatically put in a tetrahedron, and so on. And then once you connect the points, you get a simplicial complex. And you can compute, of course, just the homology. And the first the zero homology gives you the number of connected components. And of course, it's a whole sequence of homology groups. So for each radius, you get a different homology. And of course, when you make the radius bigger, certain components start connecting together, and this is visible in this diagram. But if you're interested, let's say, in the number of holes, you can compute the first homology. So here is an example. So for this set of data, so for this set of points, we can see that you get a circle here. So this loop generates the first homology, and it gives birth at some radius, and then it dies once it gets filled. So we have a birth of a homology, let's say rank, and a death, and then you get new circles. So you have new generators of the homology. So we have this birth and death of homology rank. So yeah, we know that persistence. So we will call this persistence diagrams, or we can plot it just on an XY axis to give birth and deaths of homology groups. We know that this is a stable operation. So if you perturb the points a bit, we know that these homology, let's say, barcodes, or these persistence diagrams where the x-axis is the birth and the y-axis is the death of the homology, we know that it's stable. So every method based on persistent homology to detect, let's say, lassos will be a stable algorithm. So the idea is relatively simple. So how to detect lassos inside proteins using persistent homology? Basically, you start with the loop, so you know which atoms form the covalent loop, the lasso loop, and then you take the tail, and and I keep losing my mouse. Anyway, so then you have a blue tail and a red tail, so the amino and the acid tail. And for each atom, you can construct first the persistent homology of just taking the atoms of the loop, and then you can add also the tail inside. Or you can add each atom one by one from the tail, and then get a whole sequence of homology groups. And we know that if you take the atom that pierces kind of this minimal disk, we would expect that the lifetime of a homology would be shorter, so it would drop, because the first homology group would disappear quicker, which is kind of obvious from these diagrams. So by detecting how to detect if a strand pierces the disk, we just take one atom at a time of the tail and compute the persistent homology. Now we have, for each atom, we have a persistence diagram, and then we can compute the distance between the diagram with the atom and the distance of the diagram without the atom. And there's, we experiment with the, so more or less you can use, I mean, you usually use either the bottleneck distance or the Wasserstein distance, and we use the bottleneck distance between it because it gave us the best results. And we can also see here an animation. So this black dot, these black points are part of the loop, and then we have two tails, so the red tail and the blue tail. And here we will show an animation where we compute the persistence diagram for each atom in the tail. So we first compute the atom that is the closest to the loops and so on. I think this is an animation. Opa. Yeah. And we can see that once we get closer to the minimal surface, this homology group drops. So the lifetime drops. So the green part is the theoretical intersection, and we can see a drop in the lifetime of the persistence diagram. So here these graphs are just plotting. So here is which atom we take. So the tail, this red tail, it's long, it's 30 atoms long. And for each atom, we compute the bottleneck distance between the loop without the tails and the loop with an atom of the tail. And we can see that this green dot, it's the theoretical intersection of the minimal surface, and this red dot is the maximum we get of the bottleneck distance. And we can see here that persistent homology nicely detects the exact position of the piercing of the lasso. So here are, I have just a couple of minutes left. So here are just two examples of these diagrams. Of course, the blue line is the bottleneck distances that we measured, but then we use a bit of smoothing and filtering just to make it nicer. So we can see that here there's an obvious piercing. We have just this kind of big cone where the distance changes. Here we have two piercings, and our methods are able to nicely detect it. And in general, comparing to minimal surface approach, we are up to, let's say, 88%. So 88%, we have true positives where our atom, the intersection point of our atom and the one computed theoretically by minimal surface is of at least 9 atoms apart. So we have a small amount of false negatives, a small amount of false positives, but in general, we are quite successful comparing this method and the one with the minimal surface. And I think this is maybe just this last slide. So the second experiment that we did was through molecular dynamics. So if we take a whole simulation of, so the thing is, the problem we want to explore is, we know that a protein folds going through this landscape, and it comes from an unfolded state, and then eventually through time it falls into the folded state. So the other experiment we did using persistent homology is to obtain a full simulation of a protein with the unknotted, with the non-lasso, and then through time modeled by these molecular forces. And at one point, the lasso appears. And so this is also data that was provided by Joanna Sulkowska by her previous experiments. And then we, for each frame in this simulation, we computed the persistent homology, and we can see that this was a particular interesting lasso because it was a supercoid lasso with 2 intersections. And our method nicely, we can see that the frame index is just the number of frames in the simulation, and we can see that we nicely detect the exact position in the folding of the protein where the supercoiling appeared. And you can see that the supercoiling appeared almost immediately. So there was no intermediate state with one piercing and then two, but it kind of pierced. It had two piercings almost instantly, which can also be topologically explained. Okay, this is just how this molecular dynamic simulation looks. So you put a protein inside. This is not the simulation from the data that I talked on the previous slide, but just another one. You model all these molecular forces, and you get then the 3D structure, the end one. And I think I will now stop my talk on this point since 1 hour has passed.
So thank you very much, Boštjan, for this very interesting talk. Very nice. Do we have any questions, please?
Yes, it's a very nice talk. Is it possible to use persistence homology to detect knotting in proteins?
Yes. I mean, there is one paper where they constructed, I'm not sure who it's from, but I remember reading it a few years ago. So they generated random knots through some method, and then they used, some of them were trivially knotted, some of them, I think I had the trefoil, and they used persistent homology to detect if it's knotted or not, and I think they were relatively successful with it, so you can use it.
So they were detecting the Seifert surface, maybe something like that?
Yeah, I don't know. I just know that people have done it and they were successful at it, but I'm really not sure about the details. But you can, yeah, generating just random knots, computing just the persistence homology, they're able to, with some certainty, I think, detect if it contains a trefoil or not.
And could you say something about the machine learning technique that you got for detecting knotting?
Okay, so the experiment we did with the machine learning, let me find it. Basically, what we did was take just something we know that's knotted and put it through a molecular dynamic simulation. So, which means that we were kind of, okay, so you take a trefoil and then you put it into this MD simulation. So you get thousands and thousands. So you get, let's say, 100,000 or 200,000 different configurations. And you are hoping that, let's say, if you have a trefoil, that you will get all possible configurations of the trefoil in the 3 space. And we did this with just knots. We did it with open knots. We did it with theta curves, and then we actually used protein data, those 3D protein data. And so basically, which means that you take a knot, let's say with 100 atoms, you take a trefoil with 100 atoms, you take a figure 8 knot, you take different classes, and you put them into molecular dynamics. So that's actually this simulation, exactly this one. You try to get the whole configuration space. This changed drastically over time, and then you can use AI to classify it. So you take 70% of all your simulation to classify it, which type of knot it is. So it can be the unknot up to this 5-2 knot. And we are relatively successful. So these are polymers, which kind of means artificially created chains inside the MD simulation. Then we have protein-like, which means that we also take the molecular forces a bit more naturally, so you cannot have like crazy angles. And so in some case, so the quality of this AI prediction varies a bit on the parameters of the simulation. So yeah.
Thank you. Yeah, that's interesting.
Do we have any more questions for Boštjan, please?
Boštjan, I have just a basic question, curiosity. You mentioned we had 2 numbers at the beginning of your talk. One number was more or less 1% of structures to be knotted. And then before that information, you gave us the information that, if I got it correctly, 78% are... I'd like to know, basically, how, in percentage of the 1% you mentioned, which knots are so-called deeply knotted,
Yeah.
and how many are shallow knots?
Yeah. So out of all known organisms now and all the DNA we have, so about 0.6% of proteins are knotted. So let's say roughly 1%. Out of that, 75% are deeply knotted. Or was
Okay.
it 80%? About 80% are deeply knotted. So
Yeah, 78, you said. Yes, 80%. Okay, is the 80% of the 1%, so to
yes,
speak.
yes, exactly.
Okay, those represent deep knots.
Yeah. Yes.
Okay, thank you.
We have any more questions, please, for Boštjan? Or if not, I would like to thank Boštjan very much again. It was a very nice talk. So thank you.
Thank you very much.
Thank you.
Thank you.
Thank you.
Good for you.
Thank you. Thank you.